I guess we can get started. So today we're having another session of our physiology webinars, and we're talking about the physiology of the cerebral cortex and white matter of the cerebrum. So before you get started, we're going to have a presentation by Mr. Xavier Bakasa. So before you get started, can I just allow everyone to just introduce themselves so that we can just be aware of each other and be acquainted. So I'll start with Mr. Kondwani Siame. Can you just introduce yourself? Just unmute and introduce yourself.
Good evening, everyone. I'm Kondwani Siame, a fifth-year medical student at the Copperbelt University Kitwe campus or Kitwe Teaching Hospital. Thank you.
Thank you, Kondwani. Can we have Mokaya Nyauma introduce yourself? Just unmute.
Yes, hello everyone. My name is Nelson Mokaya Nyauma, and I'm a medical student from University of Rwanda. Thank you so much.
Thank you, Mokaya. Can we have PL Panther? Hope I pronounced that correctly. PL or Pile Panther.
Okay, thank you. Good evening, everyone. My name is PL Panther. I'm a second-year medical student, Moi University School of Medicine, Kenya, and I'm a member of AFAN. I'm happy to be here today. Thank you.
We're happy to have you, PL. We also have our Director of Operations, Mr. Ebenisha Majata. Can you just introduce yourself, please?
Greetings, everyone. I hope everyone is doing well. So just as introduced, my name is Ebenisha Chonya Majata. I'm the Director of Operations at AFAN. I'm a fifth-year medical student from Copperbelt University School of Medicine here in Zambia. And I'm happy to be here. Thank you.
Thank you, Ebenisha. And in our midst, we also have our President, Ms. Lorraine. Can you introduce yourself, Lorraine?
Hello, everyone. As introduced, Lorraine Sobopelo, and I'm the president for the association. I'm really glad to be here.
Okay, thank you, Lorraine. And before we get to the presenter for today, I'll just introduce myself as well. I'm Nawantla Sakewa. I'm a member of AFAN from Botswana. I'm a recent graduate of MBBS, and I'm also happy to be here, I must say. So we're in the midst of our presenter here, Mr. Xavier. So can you please start by introducing yourself before you get to your presentation, and then go ahead and get started? Yeah.
All right, thank you very much. My name is Xavier Bakasa, fifth year medical student here in Zambia, Copperbelt University. Yeah, so my topic of discussion with you today is physiology of the cerebral cortex and the white matter of the cerebrum. And I would want to make this discussion as interactive as possible. So please tag along, and where we are not clear, feel free to ask questions. It's a long presentation, so I will try by all means to be quick with my explanations. Yeah, so where you are not clear, feel free to ask. Yeah, thank you. So let's just dive in and get started with our presentation. Right. Is my slide moving?
Yes, sir.
All right, sure. So this is the structure of our discussion today. The outline, we'll look at the terminologies in a neuroanatomy of the cerebrum and the cerebral cortex. Then we'll look at the embryology on how the brain develops, and look at the anatomy and physiology of the cerebral cortex. Then look at the white matter. Then finally we'll look at the upper and lower motor neuron lesions. So under neuroanatomy, the first term that I would want us to understand is when we say the neuraxis. So the neuraxis is just the imaginary line which is drawn through the spinal cord up to the front of the brain. So it's more like it divides the body into two halves, from left to right side. So it acts as a line of reference to the directions of a location of a particular part of the body. So the anatomical directions that we have in relation to the neuraxis, we have, when we say anterior or rostral, we mean towards the head. And when we say posterior or caudal, we mean towards the tail. Then ventral or inferior, we mean towards the belly. And dorsal or superior, we mean towards the back of the head. So just in case I use some of these terms, you'll be able to understand what I mean. Yeah, and in terms of location in the brain itself, when we say ipsilateral, it means same side of the brain. Then contralateral, it means opposite side of the brain. So those are just the basic neuroanatomical terms that I wanted us to be kept abreast with before we continue with the presentation. Yeah, so before we can even discuss the cerebral cortex itself, I would want us to understand how it develops, starting from the embryology itself. But I wouldn't want to go ham on it. I will just concentrate on areas which is of importance to this discussion. So under the development, we'll talk about the formation of the neural tube and the differentiation of these neural tube into three primary vesicles, which will form different structures of the brain. So how does the neural tube come about? So formation of the neural tube is what is called neurulation. The reason why I want us to start on the neural tube is because it's the structure in embryology that brings about the central nervous system. And under neurulation, there are typical terms that are important to neurulation. And these are the neural plate, the neural groove, the neural tube, and the neural crest. So during the development, like from first week of development of a child, or of a fetus, from first week to third week, it brings about the formation of the three germ layers, which are the ectoderm, the endoderm, and the mesoderm. So when you look at this structure here, it's in the shape of a ball, but it's more like it was a cross section. It was cross sectioned. Then this is how it looks inside. This is typical of a third week fetus, and it has three layers: the ectoderm, mesoderm, and the endoderm. But of great importance, I want us to look at this notochord, which is just embedded within the mesoderm. So how does this notochord come about? So I would want us to look at this structure here. So this is just the dorsal aspect of the bilaminar disc during the first 2 weeks of development. There is what we call the primitive. Can you see my pointer where it is, how it's moving?
Yes, we can see your cursor.
All right, sure. So this part here is called the primitive streak. The primitive streaks, it develops in the same third week, and how it develops is it develops by proliferation of the ectoderm. When it proliferates, then it forms this primitive streak. It is located on the caudal aspect of this embryonic disc. Then, during further development, at the tip of this primitive streak, that's where now the notochord comes in. This notochord is formed by further proliferation or movement of the ectoderm cells on the primitive knot, which is the tip of the primitive streak. These cells, they move in all directions of this bilaminar disc, this embryonic disc. They move rostrally and caudally in all directions. So, its development is by proliferation of the ectodermal cells. So when the notochord forms, it will be just below the ectoderm. And when the notochord forms, this is now what is going to stimulate the formation of what? The neural tube. So during the late third week, the neural tube now will start proliferating on top of the what? The notochord. And how does this same neural tube come about? So the cells are just above the notochord, they will start folding. And I think we have to understand these terms first: the neural plate, the neural groove, and the neural crest. So the neural plate cell, the neural folds are these cells which are just on the sides of this neural groove. So in between here we have the neural groove, whereas in it, the ectodermal cells, they form a groove, and on either sides of the groove, the cells, they heat up, more like they proliferate to form folds. And when they form the folds, these three structures, the neural fold, the neural groove, they are now what is collectively called the neural plate. So this structure, the neural folds and the neural groove, they form the what? The neural plate. So during development, the neural folds, they proliferate towards each other. They're more like they fold, forming a tube. And that tube, when it forms, it's what is called now the neural tube. But the cells which are on the tip of the folds here, they don't take part in the formation of the tube. They are released on the sides to form the neural crest cells. And these neural crest cells now are what will form the spinal nerves and other peripheral nervous system. Yeah. So when the neural tube now forms, it ends, the development of the neural tube, it ends around the 28th day of development. So when the neural tube now forms, at about the 28th day of development, there will be some changes that will happen, that will take place along the neural tube. So on the rostral end of the neural tube, there will be three swellings, which are called the three primary segments or the three primary vesicles swelling. And on the caudal part, it will remain as a tube. So these three segmented bodies are what are referred to as the three primary vesicles of the brain. When these three swellings, so the most rostral one is the forebrain, then the middle one will form the midbrain, and the caudal one, the most caudal one is the hindbrain. So these are called the primary vesicles. They'll give rise to other structures which will be called secondary vesicles, and from the secondary vesicles will emanate structures of the brain now. So this is just the primary vesicles. So this is just an overview of how the brain forms. So they come from the neural tube. So the forebrain will give rise to two secondary vesicles. And these two secondary vesicles are called the optic and the telencephalon. The telencephalon, which is a secondary vesicle, will give, the forebrain, which is the primary vesicle, is also called the prosencephalon. So the prosencephalon would differentiate to form two secondary vesicles, which are the telencephalon and the diencephalon. These will come from the prosencephalon. So the telencephalon and the diencephalon, these are secondary vesicles. So from the telencephalon, these will give rise now to what? The cerebral hemispheres, the basal ganglia, the hippocampus, and the amygdaloid bodies. Then on the diencephalon, this is just more like the remnant of what? The remnant of what? The diencephalon. These are also so-called the non-paired structures of the what? The remnants of the prosencephalon. So from the diencephalon, we emanate the what? The thalamus, the hypothalamus, the subthalamus, and the what? The epithalamus. So then, from the midbrain, which is a primary vesicle, will give. So the midbrain will just remain the way it is as the midbrain. It will never give rise to any structure. But there will be just other developments that will take place on the what? The midbrain, and this is formation of the what? The tectum and the tegmentum. We know the functions of the tectum and the tegmentum. They just help to move the eyes in the direction of what? The eyes, in the direction of so the tectum, it helps to move the eyes in the direction of a stimulus, and the tegmentum just helps to move the eyes in the direction of the sound. So that's just what forms on the midbrain. Then on the hindbrain, it will give rise to the what? The metencephalon and the mylencephalon. So these are the secondary vesicles. Then from the metencephalon, we develop the what? The pons and the cerebellum. Then the myencephalon will form the what? The medulla oblongata. So this is just an overview of how the brain develops. Then, as the development continues, and we know that the embryonic development, there is a lot of folding which occurs. So as there will be more folding, this part of the spinal cord or the medulla oblongata will be moved dorsally, and this part will move ventrally. So the combination or the aggregate of such movements will form this structure of the brain. You'll find that the cerebral cortex will move dorsally and caudally. Then the medulla oblongata will move ventrally and rostrally. So that aggregate of such folding will form the brain structure, which looks something like this. So the more like the cerebellum will overlie all the structures, and these other structures just lie below it. Yeah, so I think what I've explained, everything is on this slide. And this flowchart is just trying to explain how the three primary vesicles, they differentiate into the secondary vesicles and giving rise to the structures that we mentioned. Yeah, so differentiation of the forebrain, I think I've mentioned that. So now the topic of our discussion today is mainly, we're not interested in these other structures, but we're just interested in what is happening in the cerebral cortex and its associated white matter. So the forebrain structures and the function relationships. The forebrain is the seat of perception, conscious awareness, cognition, and involuntary action. So it's more like it's called the higher center of the brain, because that's where most important functions of the brain lie. It controls almost each and every part of the brain, and most of the important functions are consciousness and cognition, voluntary action, personality, even emotions. They are all controlled in the cerebrum, and it's a very important structure to look at. So when we partition the cerebrum itself, the cerebrum is the largest division of the brain. So it's divided into two hemispheres, and each hemisphere has 4 lobes. So the cerebellum is gray in color because it's formed of cell bodies. So that's what characteristically give it a gray color, because of the cell bodies of the neurons, the brain cells. So when we have a cross section of the brain or the cerebral cortex, this is how it looks from the inside. So the outer margins, they look gray because of the cell bodies, and this gray matter is what. So the cerebral cortex is just initially the gray matter. So it forms the superficial aspect of the cerebellum. The cerebrum. Then this whitish part just under the gray matter or the cerebral cortex is what is called the white matter. So this white matter is made up of what? The axons now. They transport information from the cerebral cortex to other parts of the brain and to the rest of the body, as we'll see along the discussion. So the cerebral cortex, it has some contours and surface features that are of great importance to us. So on the cortex, the surface is more like it has more like convolutions or elevations and grooves. So these grooves are called sulci and fissures. So the small grooves are called sulci and the large grooves are called the fissures. Then the bulges between the fissures and the sulci are called the gyri. So as you can see from here, it looks more like intestines. They are compacted on each other. So these elevations, these are the same. These are the gyrus I was talking about. Then these small grooves are called the sulci. Then these large grooves, like this one here, they are called fissures. So that's basically the anatomy or the surface anatomy of the cerebral cortex. So, and the cerebral cortex is composed of cells. There are more like 6 layers of cells in the cerebral cortex. I didn't delve much in that because I thought it would take us the whole night. So it's just that the cerebral cortex has 6 layers of cells, and these are what gives the cerebral cortex its typical functions. So from the cerebral cortex, this slide is just trying to explain the number of neurons in the cerebral cortex is estimated to be 10 to 10 billion, and there are no basically gender difference in the cortical thickness, but some studies say the density of the cerebral cortex is higher in men, with a reciprocal increase in the neural pure and neuronal processes in females. So more like there is more of a cerebral density in males than females. So I don't know. Maybe that's why you are the head of the house, something like that. So the cerebral cortex, these features, I already mentioned them, the gyri and the sulci. So, and of importance, from the sulci, we have the central sulci, which divides the frontal lobe from the parietal lobe. This is an important landmark, the central sulci, because it divides these 2 important lobes, as we'll see the importance of this central sulci. Then we have the fissures, which are the deep grooves. So these deep grooves, they separate mostly the lobes on the same side of the brain or ipsilateral side of the brain, and So under fissures, we have 3 important fissures. So we have the longitudinal fissure, which divides the two cerebral hemispheres. Then we have the transverse fissure, which separates the cerebellum from the cerebrum. Then we have the sylvian or the lateral fissure, which divides the temporal lobe from the frontal and the parietal lobe. So this is the lateral fissure or the Sylvian fissure. It separates the temporal lobe from the frontal lobe and the parietal lobe. So the central sulci is somewhere here, the central sulci. So this separates the frontal lobe from the parietal lobe. So these are just diagrams showing the anatomical relation of those fissures and the sulci. And this is just a longitudinal section to dive deep on how the 3D orientation of the sulci, the central sulci, we can appreciate it, how deep it goes, and these other fissures and sulci. So we have general functions of the cerebral cortex. So the general functions of the cerebral cortex, it's a center of perception. Perception of fine sensations. Yeah, fine sensations to say. Like fine sensation, what we mean by fine sensation is a touch, fine touch, and other perceptual stimulus like sound and the like. So it's also involved in the initiation of skilled voluntary movements. This is important because skilled voluntary movements, you know that sometimes people can have a learned skill like riding a bicycle, but they don't forget, even if they rode a bicycle like 20 years ago, but still they can ride. Why? Because that was the skill was learned and it was kept in the cerebral cortex. And of great importance again is those who play piano, guitarists, violinists. They all have those skills because they built the cerebral cortex. It learned that skill and kept it. Then it's also involved in consciousness. We are all aware that we are alive because of the cerebral cortex. But there are some people who believe that they are dead. So I don't know the function of the cerebral cortex. Maybe it's dead as well. But yeah, there are some people who do believe, especially those who have psychotic conditions. They believe they are dead. So maybe the problem can be with their cerebral cortex. So it's also called the high intellectual functions. It has it houses high intellectual functions like speech, learning, and memory, as we'll see from the few slides to come. So as we alluded earlier, we said the cerebral cortex is divided into partitions or lobes. So each lobe has got different functions and different effects on the human body. So these lobes, they are more like we have the same number of lobes on each cerebral hemisphere. We have the frontal lobe, the parietal lobe, the temporal lobe, and the occipital lobe. So we'll start with the frontal lobe, which is color-coded blue here. This is the frontal lobe. So it's separated from the parietal lobe by this central sulci, and it's separated from the temporal lobe by this lateral fissure or the Sylvian fissure. So the frontal lobe, it's located deep to the frontal bone of the skull. We know where the frontal bone is. Then it consists primarily, it consists of the primary motor cortex, the motor association cortex, the frontal eye fluids, prefrontal cortex, and the Broca's area. So this is just the diagram trying to highlight those structures, how they are oriented on the cerebral cortex, on the frontal lobe. So the area here in blue is the primary motor cortex. And what's the function of this primary motor cortex? It's also called the precentral gyri. Because it lies like in front of the central gyri. The central gyri is here, so it's also called the precentral gyrus. So this primary motor cortex, it is involved in what? Voluntary movement on contralateral side of the brain. Why are we saying contralateral side of the brain? We know that the message or information which emanates or originates from the cerebral cortex, it will be transported by the white matter or the corticospinal tracts, and we know that when it reaches the medulla oblongata, those corticospinal tracts will decassate or they move on the contralateral side. So no wonder the primary motor area cortices they affect the voluntary movement on the contralateral side of the body. Then, so we say the primary motor cortex is just involved in the voluntary movement on the contralateral side of the brain, or I mean on the contralateral side of the body. So like what kind of voluntary movements, like or just moving or doing any activity that you feel like doing. So it's under the conscious awareness of the cerebral cortex. So it's affected by the primary motor cortex. So of importance here is to note the word motor cortex. So it's more like the efferent effect, not the afferent, but the efferent effect. You might have received a message from the peripheries, but now a feedback from the cortex to that stimulus will be propagated by the primary motor cortex. Then, just after the primary motor cortex, we have what is called the motor association cortex, which is color-coded red here. Now, the function of this motor association cortex, it's more like it keeps memory of the movements. Like I said earlier, when you learn a skill, like riding a bicycle, you don't forget after so many years, because that skill, that voluntary action or that movement, the compound of those movements was what? Kept in the what? Motor association area, such that next time when you go back to that activity, you are able to play it with or you are able to do it with preciseness because of what? The motor association area. So it just complements the function of what? The primary motor cortex. So it is involved in what? Recognition. It's involved in planning, sequencing, and execution of movements. So this motor association cortex is also called the premotor or supramental motor cortex. So don't get confused when I say premotor or supramental cortex. So the motor association cortex, it complements the function of the primary motor cortex. Then we also have the frontal eye fields, which are involved in movement of the what? The eyes. Then we have the prefrontal cortex. So on the prefrontal cortex here, this part is more like, it's very important. Why it's important? Because there are very cardinal functions it holds, like memory, and of great importance is personality, reasoning, even decision making, learning, and behavior. So these are higher functions, such that when there is a lesion in the prefrontal cortex, these these functions will be affected. Then on the prefrontal cortex, this cortex also, it sends fibers to the motor association cortex, which is involved in planning of movements. So we'll see how that coordinates when we look at how a movement is brought, how a voluntary movement is brought about. Then, of importance again on the frontal lobe, we have the Broca's area. So this light blue here, so the orientation, I found it difficult to arrange these things, but hope it's clear. So this part here is the what? The Broca's area. And this Broca's area, it is located on the dominant cerebral hemisphere. you won't find the Broca's area on both sides of the cerebral hemisphere, but it's only located on the dominant hemisphere of the frontal lobe, of the cerebral hemisphere. So mostly those people who are right-handed, their dominant hemisphere is the left cerebral hemisphere, and those people who are left-handed, their dominant cerebral hemisphere is the right hemisphere. So it's this Broca's area. It's located inferior, and it's located inferior to the frontal lobe, and just near the prefrontal cortex. And the function of this Broca's area, it is involved in speech production and language comprehension. So it also controls the facial neurons. So this function is very much important in that for us to speak like the way I am fluent, the way I'm speaking fluent, it's because of this area is functioning. But if there was a lesion in this part, like the Broca's area, there will be a condition which will be known as the Broca's aphasia. So this Broca's aphasia, it's brought about, by definition, it's more like for simplicity's sake, it's just the inability to be fluent with constructing sentences. Like, for example, someone with Broca's aphasia, they won't construct a full sentence. Like, for example, for a sentence like, I'm going to the market, but someone with Broca's aphasia won't construct that full sentence. They'll be like, I market going. Like, there'll be shortage of the prepositions in those in the sentence to make it grammatically correct. More like it will be missing. Why? Because of Broca's aphasia. So it results in the inability to they comprehend. Yes, they can listen what you are saying and understand it, but now for them to give feedback, it's very difficult because of the lesion in this area. And these people, like Broca's aphasia, it can be caused by maybe a stroke in this area, like if there's a stroke, and or a thrombi in the vessels which supply that part of the brain. And the thrombi, mostly it can occur in the middle cerebral artery or the internal carotid artery. So if there's a thrombi in those vessels, then it's likely that they will affect this area, the Broca's area. Even infections also, they can affect the Broca's area. But of great importance is that you can see how it's located, like it's just near the prefrontal cortex. So it's inevitable sometimes to find that a lesion in here will also affect the prefrontal cortex. No wonder some studies say people with Broca's aphasia, they are more like irritable, like they have more like short tempers, because sometimes they say it can be attributed to them failing to talk nicely or to construct a full sentence. And sometimes maybe the lesion could have also extended to affect the prefrontal cortex. And we saw that the prefrontal cortex is involved in what? The behavior and even reasoning. Sometimes you find that these people, they can somehow be cold-hearted, Yeah, so it's because of maybe the lesion it extended to affect this area. So it's important to know that the Broca's aphasia, it affects the Broca area, and these people, they will find it very difficult to be fluent with to construct a grammatically correct sentence. Yeah. So this diagram, it shows the primary motor cortex, like the cross section of the primary motor cortex. As you move from this part here, it's more like that's where there is a longitudinal fissure which separates the two hemispheres. So maybe this can be the right hemisphere, and here there could be a left hemisphere. So by just zooming into this part, as you move laterally to as you move towards the temporal lobe, the different areas of the primary motor cortex, they affect the movement or the function of different areas of the body. So as you move from the midline here, going laterally, you find that it's on the most medial area. This area, it controls the lower limbs, and as you go laterally, it controls the upper limbs, and most lateral towards the temporal, it controls the function of the head. And of interest here, you can see that the hand looks big and the head looks big, but the limbs, they look small. This is just a representation, a graphic representation, to let us know that the size of the limbs or the part of the body is proportional to the area of the primary cortex that controls it. So the hand here is big because it has a larger proportion of the primary cortex which controls the movement of the hand and the head. So this is a typical of a homunculus. A homunculus, yeah, where it has disproportionate parts of the body. It has a big head, big hands, small trunk, and small legs. Yeah, so that's typical of a homunculus. So now, just to appreciate the function of the frontal lobe, we can see like how planning and execution of a voluntary movement comes about. So the planning, from our discussion, we saw that the planning comes from the motor association cortex. So when the plan comes from the motor association cortex, that plan is sent to what? The basal ganglia or the lateral cerebellum. So now, what would determine what route to take for that plan is the type of movement that will be needed after that plan is executed. For example, if a fast response is needed, or when an idea is formed, then a fast movement is needed. So that information from the motor association cortex, it will be sent direct to the lateral cerebellum, because from here, that's where it comes in, like fast movements. Then if it is a slow movement, that idea will be sent to the basal ganglia. Then from there, like from these two centers now, that idea is screened. Then they say, okay, let's see what we have here. So this is the idea. Okay, so the plan of a movement will be like this and this and this. Then when that plan is formed, it will be sent back now to the primary motor cortex. So what I'm saying is the motor association cortex will form a plan. Then from here, it will be sent to the basal ganglia or the cerebellum. Then when that plan is formed, then it will be brought back to the primary motor cortex here. Then when the plan of movement is brought to the motor cortex, primary motor cortex, then now for it to be executed, the impulses will be sent to the specific part of the body which needs to be moved. Let's say the hand or the leg. So it will be moved to the plan, or the plan to execute that movement will be transported to that limb via the corticospinal tracts, which are the white matter, as we will see. So this is just a brief summary of how a plan and execution of a voluntary movement comes about. So this slide now, it just tells us about. So it's an experiment which was done just to appreciate the function of the motor association cortex, the prefrontal cortex, and the primary motor cortex. So in this experiment, there were two things that the participants were told to do. So they were told to learn a particular movement of a pattern in the fingers. So once they learned that pattern of movement in the fingers, then their electrical activities was monitored, and their cerebral blood flow was measured. So of interest, as the person was executing, so they were first told to think about the finger movement, pattern and then to execute that movement. So as the experiment was going on, they realized that most of the brain activities was happening in the primary motor cortex and the premotor cortex, or the motor association cortex, and even the prefrontal cortex. That was when the participants were moving the fingers in a pattern. But then the participants now were told not to move the fingers in a pattern, but just to think about that pattern. So when that was happening, they just realized that most of the brain activity and the cerebral blood flow was in the premotor area and the prefrontal cortex. So this showed that the premotor area, or the motor association cortex, and the prefrontal cortex, these were involved in planning or thinking about something, but the actual execution was manifested in the primary motor cortex. So this was just to appreciate about the functions of the motor association cortex and the primary motor cortex. So then we look at the parietal lobe. So the parietal lobe is post-central. It's on the post-central gyrus. So the post-central gyrus is somewhere here. Oh, it's here, actually. So this green area here is what is called the parietal lobe is the whole part, this light green. I don't know. I didn't do colors at juniors. So I don't know if this is apple green or white and the like, but this area, which has more like a green, it's what is called the parietal lobe. So on the parietal lobe, we have different structures which form the parietal lobe. So the parietal lobe is located in the parietal bone of the skull. The skull. So it consists of the primary somatosensory cortex, the somatosensory association cortex, and the posterior association area. So this part here is what is called the primary somatosensory cortex. Then this area here is what is called the somatosensory association cortex. Then I forgot to put the color code for the posterior association area, but it's somewhere located here. So the function of the primary somatosensory cortex is involved in awareness of somatic sensation. And these somatic sensations are what? Fine touch, proprioception, and vibration, such that when someone touches you, you'll be able to take, okay, this is a touch from a lady, and this is a touch from a guy, because of your primary somatosensory cortex is functioning. But if your primary somatosensory cortex is not functioning, then you never distinguish from a fine touch and crude touch. So somatosensory association cortex now, it's just involved in the analyzing of a touch and recognition of the somatic sensations. When someone touches you, okay, you know that, okay, this is a touch from a person. You've recognized and analyzed that because of what? The somatosensory association cortex. So I want us to look at the pattern which is happening here. For each and every lobe, if we have a primary area, like in the frontal lobe, we had a primary motor cortex, and we had also an association cortex to primary motor cortex. Even here in the parietal lobe, we have a primary somatosensory cortex. So we must also have what? An association cortex to that. So mostly the association cortices, they are involved in what? Analyzing and recognition, and also planning for what follows next. Then we have also the posterior association area. So this posterior somatosensory area, it's a multimodal association area. So it coordinates the association areas from the visual, the auditory, and the sensory areas. So it's a multimodal association area. It's connected to these other association areas. So I can give an example of how this posterior association area works in a situation where, as in, for example, you see a lion. It roars at you. So from there you have three stimulus which is happening. You see it and you hear it. So all those stimuli, they'll be coordinated here in the posterior association cortex to bring about the plan of what follows next. If you are going to run or you are going to fight that lion. So it coordinates those stimuli to make you recognize, okay, that's a lion. And then that posterior association area is connected to that, the primary motor cortex here in the frontal lobe, so that it helps you to plan on the type of a voluntary movement which will be required thereafter, if it's to run or to fight. So that's all about the about the parietal lobe. So we have now the temporal lobe. So the temporal lobe is located on the sides of the brain, deep to the temporal bones of the skull. So it consists of the primary auditory cortex, the auditory association cortex. Again, I guess we are now aware of this pattern of a primary and an association cortex. So we have a primary auditory cortex and an auditory association cortex. And I guess we can even know that the primary auditory cortex is involved in conscious awareness of the sound stimulus. And of importance that this primary auditory cortex, it responds to pitch and frequency of sound and the location of sound. Okay, you'll be able to tell, okay, this sound is coming from this area, and it will be able to tell, okay, this sound is loud and this sound is soft. So those sensations will be picked by the primary auditory cortex. Then the auditory association cortex, these now will analyze the sound. So it helps to recognize the sound. Is it a voice or... So, for example, if you are living with someone, there are some times where when you just hear the voice, you'll be able to tell, okay, that's a voice of John and that's a voice for who, because you know voices. And their sound is what? It's kept in the auditory association cortex, because their sound was kept in the association cortex. So you'll be able to analyze that, okay, that sound is a sound of this, because the memory was kept in the association cortex. In the other area on the temporal lobe, sorry, I didn't show the temporal lobe. So this is the temporal lobe. Then it has the Wernicke's area and the primary auditory cortex. So the auditory association cortex just here, like, didn't know how to color code it, but they just lie more like the Wernicke's area is medial to what? To the auditory association cortex. So the Wernicke's area, just like a Broca's area, it is a language. It also involved in language and speech and comprehension. So this area, it's more involved in comprehension of speech, of language comprehension, like a heard speech or a written one. So this area, it controls for us to understand that, okay, what this person is talking about is this, because the Wernicke's area is functioning. You are able to comprehend what they are saying or what they are writing. But a situation can occur whereas a person can have a lesion in the Wernicke's area. So, and that will lead to what is known as the Wernicke's aphasia. So, in these people with Wernicke's aphasia, they can't comprehend, like, they can't understand, like, the language. They don't understand because the comprehension area is affected. So words and sentences are not clearly understood, and sentence formation is also inhibited. So that's all about the Wernicke's area. Then, of also importance in the temporal lobe is what? The primary olfactory cortex. So this olfactory cortex is what? Involved in the awareness of what? A smell. So the primary olfactory cortex is located somewhere here. So it's involved in the awareness of smell. Then you also have olfactory association area. I guess we know what that means and their function. It analyzes the smell to know, okay, this is a smell of rotten things, and this is a smell of a good fragrance and like. So it's because of the olfactory association cortex. Then there is also some fibers, or white matter fibers, which run from the temporal lobe to the inferior part of the frontal lobe. And these are called the arcuate fasciculus. So this is just a white matter tract that connects the Broca's area and the Wernicke's area through the temporal lobe and the parietal lobe. As we can see here, so the Wernicke's area and the Broca's area, they are separated by this Sylvian fissure. So these areas, they are called parasylvian speech centers. That's the collective name they are called because they are located on either side of the Sylvian fissure. And between these two structures, what connects them is what? These white matter fibers called the arcuate fasciculus. Such that when someone has a lesion in here, it is what we will be referred to as the conduction aphasia. So, of great importance here to how you know that this is a conduction aphasia. You find that this person will comprehend the language, to understand, and they will speak nicely, but they'll have difficulties repeating some phrases. Why? Because the conduction here is affected. They'll understand and they'll speak, but for them to repeat what you are saying, it becomes a problem because of a disconnection between these two areas. So we also have the occipital lobe. So this occipital lobe of the brain is located deep to the occipital bone of the skull. It consists of the primary visual cortex and the visual association cortex. So it consists of the primary visual cortex and the visual association cortex. Then the primary visual cortex, obviously, it's involved in conscious awareness of visual stimulus. Then the visual association cortex, it just analyzes and recognizes and interprets the visual stimulus. Okay, to say, okay, what I'm seeing is a pen, what I'm seeing is a ruler, what I'm seeing is a marker. So it helps you to analyze and recognize that because of what? The visual association cortex. So this is the primary visual cortex, and an association cortex is just right next to it. Then I think the insular lobe, I didn't mention it on the types of lobes that we have. I think the reason why I didn't do that is because on the surface anatomy, you won't appreciate the insular lobe. So the insular lobe, it's located deep to the temporal lobe. So when you put like hooks here on the Sylvian fissure, then you separate the frontal lobe and the temporal lobe. So this area here, that's where the insular lobe is located. And so here on the text it says it's located medial to the rectosulcus or the Sylvian fissure. So it consists of the visceral sensory cortex, gustatory cortex, and the vestibular cortex. So visceral sensory cortex is just a conscious awareness of pain coming from the viscera, like the GI. If you have stomach pains, the sensation from those pains will be coordinated in the insular lobe. And also gustatory cortex, it's involved in the awareness, tasting things, like you taste something bitter, sweet. The cortex is involved in the gustatory cortex is located in the insular lobe. So, and on these cortices, like the visceral sensory cortex, you have the association cortices. So gustatory cortex also have gustatory association cortex. which helps you to recognize, okay, this is a taste of what, something sweet. This is a taste of sugar, and this is a taste of salt. Why? Because your gustatory association cortex is functioning. You'll be able to distinguish the different tastes. And you also have the vestibular cortex. So this is just awareness of rotational movement and static equilibrium. When someone is rotating, so this vestibular cortex, it will help you to coordinate and be conscious of that rotation. You'll be able to tell that, okay, I'm rotating clockwise. So for you to be conscious aware of that is because of the vestibular cortex and the vestibular association cortex. So I think that's what I had for the lobes and the cerebral cortex. So if to this end there is any questions, please let me know. You can just post your questions in the discussions, then I'll be able to respond to them later. Or we can put or we can be discussing in the inbox. So this picture here, it's just to help us to gauge of how much we've gotten from this presentation. So, like, we have a group of I don't know if it's neurosurgeons. Yeah, they are neurosurgeons. So they are they poke a certain area in the brain, and of interest that the limbs, it moves up, like it jerks up, as you can see here. So, from what I've just presented, what area of the brain? The area. So when I say the area, you have to tell me the lobe and the specific area that these surgeons are stimulating for them to have this jerky movement. When they poke on that area, then the limb will go up. Yeah. So we'll get back to that. So anyone with an answer can just send.
Can I give that now?
Yeah.
So this area which was stimulated, it was a middle side. Because when we are talking about the homunculus, so the legs and now from the foot to upwards is begin the middle side of the cerebrum.
All right. Yeah. So they are Sorry. Right. I think I've lost a lot of people. I'm only seeing 3 people online. Are we together?
Yes, is my answer clear?
All right. Please, can you repeat your answer? I didn't get it.
Okay, I'm saying the area which was stimulated, it's the middle of the cerebrum, because when we are looking at the homunculus, so now it's begin from the middle side, so the face is going to be in the lateral side, which means now the foot is let's say it's extended, so we may say that it was a somatosensory area which from the middle side was stimulated by the surgeons.
All right, thank you for that. Sorry, they are stimulating the primary motor cortex and specifically on the medial side, just near the longitudinal fissure of the brain. Yeah, so this is a scenario of a scenario. So I don't know if because I think we still have some slides to talk about, but I'll just read through so that we just consolidate and appreciate the previous discussions we had. So this scenario is about this gentleman, Phineas Cage, who was a railroad worker in the 19th century, living in Cavendish, Vermont. So one of his jobs was to set off explosive charges in large rock or in order to break them into smaller pieces. So one of these instances, the detonation occurred prior to his expectation, resulting in a 42-inch long, 1.2-inch wide metal rod to be blown right up through his skull and out through the top. So it entered from here to here. So the rod entered his skull below his left cheek and exited after passing through the anterior frontal lobe of the brain. So this is how the rod entered. Then, remarkably, Gage never lost consciousness or quickly regained it. There is still some debate on this. So he suffered little to no pain and was awake and alert when he reached a doctor approximately 45 minutes later. So he had a normal pulse and a normal vision, and following a short period of rest, he returned to work several days later. So, yeah, this is just way too much human comprehension. You can't understand this. However, he was not unaffected by this accident. He was just normal. So, something which is of debate until now. So, recording what you have learned regarding the frontal lobe, what possible problems or abnormalities may Gage have presented with subsequent to this type of injury? So we have to remember the precise location of the rod through the brain. So what are some of the possible problems and abnormalities that can present from this kind of an injury? So you can just type in, or you can just unmute your mic and give a fast response. Okay. So for this scenario, I think we'll get back to it once we are done with everything. But just wanted to give you something to think about. But obviously, just to give us an idea, this is entering through the inferior part into the frontal lobe. And mostly, the most affected, the part of the frontal lobe which was most likely affected was what? The prefrontal cortex. And from there, then we can know that what type of abnormalities Gage would have had later in life. So these are the answers to that. So we'll come back to it. But I want us to rush through the white matter before, because I'm looking at time. White matter, when we say white matter, we just mean, I can just say these are just axons or fibers that run from the brain and connects the different parts of the brain and to the rest of the body. So they are more like tracts of fibers. So they are found in deeper tissues of the brain. They are subcortical. So it contains nerve fibers, which are extensions of the nerve cells from the cerebral cortex. So many of these fibers, they are surrounded by a sheath called myelin. So this gives it the white color. So the difference between gray matter and white matter is that the gray matter, it occupies about 40% of the brain tissue. Then the white matter has about 60% of the brain tissue. And the gray matter, it contains cell bodies, and the white matter, it contains bundles of fibers that run from the brain to all the way to the limbs through the spinal cord. Then the gray matter, it processes information, and the white matter, it relays that information from the cerebral cortex to the rest of the body through these tracts of fibers. So white matter has 3 types of fibers. These tracts, so we have commissural fibers, projection fibers, and association fibers. So when we talk about commissural fibers, just mean those fibers that connects 2 two sides, opposite sides of the brain, like two opposite sides of the hemispheres. They help to coordinate their function. So that it relays information from this cortex, from this hemisphere to this hemisphere, to help coordinate their function. So main commissural fibers are the corpus callosum, the anterior commissure, the posterior commissure, the abenula commissure, and the hippocampal commissure, the forceps minor and the forceps major. So this is just the dorsal view of the brain. So this is So this is the right hemisphere, right cerebral hemisphere, this left cerebral hemisphere. So you can see these fibers which are running transversely. So these are the commissural fibers. They help to coordinate the functions of the two cerebral hemispheres. So commissural fibers, we have types. We have the corpus callosum, anterior commissure, posterior commissure, the abenula commissure. I've mentioned these. So from the location of these, like for example, the corpus callosum, it connects almost each and every corresponding areas of the two cerebral hemispheres. So it runs from here to here and helps to coordinate the functions of these two cerebral hemispheres. Then again, the anterior commissure, it connects structures of the olfactory pathways on the frontal lobe, the temporal lobe, and the parahippocampal area. So when you look at this slide, you'll be able to understand how these commissures, they coordinate the function of these opposite sides of the cerebral hemispheres. Then we have association fibers. Association fibers, these they connect lobes on the same side. So they don't involve. They are not involved in connecting different lobes, but they coordinate functions of the lobes on the same side of the hemisphere. So we have two types of association fibers. You have short association fibers and long association fibers. So short association fibers, they connect adjacent gyri, the same lobes, like from here to here, they're short association fibers. But we have long association fibers, and a good example is the arcuate fasciculus that we mentioned, which connects the Broca's area and the Wernicke's area. That's a good example of a long association area. Then we have the last fibers called the projection fibers. So these projection fibers, they run all the way from the cerebral cortex to the spinal cord. And so it consists of both the efferent and the afferent fibers and helps in uniting the cortex with the lower parts of the brain. So two types of projection fibers, you have corticofugal and corticopedal. The corticofugal fibers, these are efferent fibers, so they run from the cerebral cortex and terminate in the basal ganglia or the brainstem or the spinal cord. But the corticopedal fibers, these are afferent fibers, so they typically originate from the thalamus and terminate in the cerebral cortex. So these pink color-coded fibers, these are what refer to as the projection fibers. So you can see that they run from the cortex and all the way to the spinal cord. So this is a very important discussion that I want us to look at the upper and the lower motor neuron lesions. And this is to summarize everything that we've been discussing. So I hope I still have people with me online. You can be talking to yourself. Are there any questions to this end?
No questions so far. You can finish and then we'll ask questions.
All right, sure. Thank you. So before we talk about the lesions themselves, we have to understand what is an upper motor neuron and what is a lower motor neuron. So upper motor neurons, these are neurons that run from the cortex to all the way to the midbrain, the pons, and even to the spinal cord. So like from this diagram, these are upper motor neuron lesions. They run from the cortex all the way down to the spinal cord. And they terminate in the what? The anterior gray horn. So it's very important to note that. So even before we talk about that, we have to know that there are two types of upper motor neurons. So we have the corticospinal tract and the corticobulbar tract. So the corticospinal tracts, these they run from the cortex all the way through the internal capsule into the spinal cord, and they give axons to the anterior gray horn. And I think I alluded to that. Then the corticobulbar tract, they arise from the cortex and they terminate into the the bulb of the brain, which is the cranial nerve nuclei in the midbrain. So that's why it's called corticobulbar tract. So, and the damage to this tract is called pseudobulbar palsy. Why it's called pseudobulbar palsy? Because let me have something. Or like from here. So, like for this neuron, it runs all the way to here. So this area here, there's a nuclei here, the cranial nerve nuclei. So if there's a lesion to this tract from here to here without involving the cranial nerve nuclei, then it's called pseudobulbar. But if it involves the cranial nerve nuclei, then it's called bulbar palsy. So these are the upper motor neurons. So from the cerebral cortex to the anterior gray horn, and from the cerebral cortex to the cranial nerve nuclei. And there are specific cranial nerve nuclei. So starting from the cranial nerve nuclei and all those neurons that emanate from the anterior gray horn to the peripherals, that's what constitute now the lower motor neurons. So the lower motor neuron components, it contains the anterior gray horn plus the efferent axons, then the cranial nerve nuclei. So of importance here is that the cranial nerve nuclei, those which are called lower motor neurons, are the motor nuclei plus their efferents. So it's not all the cranial nerves, but only cranial nerve 5, which has a motor component which supplies the muscle of mastication, cranial nerve 5, then cranial nerve 7, which supplies the muscle of facial expression, then also the nucleus ambiguous, which is an aggregate of 3 cranial nerve nuclei, cranial nerve 9, 10, and 11, and also cranial nerve 12. So the nuclei of these cranial nerves and their efferents, they are all under the section of lower motor neurons. So what can cause damage to the upper motor neurons or the lower motor neurons? So when we start with the upper motor neurons, we have to look at the most common one, which is the CVA. So either hemorrhagic or ischemic, they can cause an upper motor neuron lesion. So that stroke can affect any along this length of this tract. So as long as it just affects any of this pathway or tract, they can cause a upper motor neuron. Then multiple sclerosis, Friedreich ataxia, and vitamin B deficiency, these will just cause demyelination of the axons. So that will affect the transmission of nerve impulses. So that will cause a dysfunction, proper functioning of those tracts. Then you also have amyotrophic lateral sclerosis. So they affect the nuclei in the cerebral cortex cells. So it hammers these nuclei. Then it means even the plan or movements or anything, all the impulses from here, they can't even be formed. It's because this lesion affects the cell bodies in the cerebral cortex themselves. Then you have the lower motor neuron lesion, the etiologies. Most important one or most common one was the viral infections, especially poliomyelitis, where it affected the anterior gray horn. But this disease nowadays is rare because there have been like interventions that have been put in place to cure this, and it's It's a rare disease right now, as in nowadays. Then you have spinal muscular atrophy, Coda equine syndrome, whereas you have a herniated disc which is compressing on the cranial nerve on the lower motor neurons. Diabetic neuropathy, which affects the transmission of the nerve impulses. Botulism. So botulism, it's more like it affects the release of the neurotransmitter like acetylcholine, such that an effect of that neurotransmitter is not felt. So that will lead to a lower motor neuron lesion, as we'll see when I explain something later. Then amyotrophic lateral sclerosis, just like this. So amyotrophic lateral sclerosis can cause both upper and lower motor neuron lesions. So a patient who is affected with the amyotrophic lateral sclerosis can have symptoms of both upper and lower motor neuron lesions. So this table just try to summarize the differences of how you can know that, okay, this is an upper motor neuron lesion and this is a lower motor neuron lesion. So there are components or parameters that we need to look at as we are trying to differentiate between an upper and a lower motor neuron lesion. The first parameter that we're going to look at is what? The mass of what? A muscle. It can be a muscle in the upper limbs or a lower limb. If someone has an upper motor neuron lesion, excuse me, they can have a decreased muscle mass. In fact, in both the upper motor neuron lesion and lower motor, they can have a decreased muscle mass. But what will help you to have a high index of suspicion that this is an upper or a lower motor neuron? So we need to understand the physiology, because if you just master, it will be difficult to use more energy to master, as you understand. So let's say someone has an upper motor neuron lesion, then there is what? Atrophy of the mass. What is causing that? So let's say, so this is just a diagram showing the corticospinal tract all the way to the anterior gray horn, then from there the efferent fibers to the muscle. So if you have a lesion on any of this tract, then it will lead to what is called the upper motor neuron lesion. So let's say, for example, you have a lesion in here. So it means voluntary. Let's say you want to flex. Let's say this is the biceps. You want to flex them. You want to flex your arm. So for you to voluntarily flex that arm, you won't be able to flex because the voluntary action of the cerebral cortex is cut off from being connected to this lower limb. So it means you won't be using this these muscles. So, and we know that if you don't use it, you lose it. So meaning these muscles which won't be used, they are not under voluntary action, action of the cerebral cortex. So they will be, they won't be used. So the type of atrophy that will occur is what? Disuse atrophy. If you don't do something, it's like to atrophy. So like mass, the type of atrophy is called disuse atrophy, and you just the muscle mass will just decrease about 15 to 20%. It won't be that much decrease in the muscle mass. So, but what will help to know that this is an upper motor neuron, the lower motor neuron lesion, is that the muscle mass will be lost drastically in the lower motor neuron. And why is it the case? Because when you look at the lower motor neuron lesion, when we say there's a disconnection in the upper motor neurons to these muscles, it didn't affect the release of neurotransmitter to these muscles. Neurotransmitters were being sent here because the lower motor neurons were intact. So that's why there was just a small change in the muscle mass. But if you look at this second diagram, let's say there's a lesion on either here here or any part along this lower motor neuron. So you find that if there is a lower motor neuron lesion, it means neurotransmitters won't be released. And an example is acetylcholine. And the function of acetylcholine, when it's released in the neuromuscular junction here, it will bind to these receptors on the postsynaptic left, like on the muscles themselves. Then it will induce some intracellular mechanisms, calcium influx. Then it will lead to contraction of this muscle. And that will be under a voluntary action. Let's say in the cerebral cortex, the impulse will be sent here to here and the acetylcholine will be released. But if there's a lesion here, it means acetylcholine won't be released here. So if there's few acetylcholine here, it means there will be no contraction. But this is not what is really causing a drastic loss in mass. We know that acetylcholine, when it's deposited here, it will bind to the nicotinic receptors and to the muscarinic receptors. But if there's no acetylcholine here, then it means these muscarinic receptors will be not stimulated. And the muscarinic receptors are very much important because they help in signaling transduction or transcription factors, which will help in the synthesis of muscle proteins, such that, or for example, those people who lift weights, because of the work of these muscles, there will be more proteins that will be formed because of the function of these muscarinic receptors. So if you don't have acetylcholine, then it means no new muscular proteins will be formed. And if no new muscular proteins will be formed, muscle proteins will be degraded. We know that there's a fine balance between protein degradation and protein formation. So if there's a decrease in protein formation, then protein catabolism will take the day, and there will be a loss, drastic loss in the muscle mass because of the degradation of the muscle proteins, which are resulted from the ineffectiveness of these muscarinic receptors. So these people with lower motor neuron lesions, you expect them to have a decreased mass, a drastic change in the muscle mass. Then the strength, when you look at the strength, I guess we know that in upper motor neuron lesions we have spastic paralysis, and in lower motor neuron lesions we have flaccid paralysis. But what does this mean? How does this come about? So for us to understand spastic paralysis and flaccid paralysis, we have to look at the muscle tone and the deep tendon reflexes. So the muscle tone is affected by the muscle fibers. So on this lower motor neuron here, if you look at this diagram, this efferent lower motor neuron, it has two types of fibers. You have the alpha motor fibers and the gamma motor fibers. The alpha motor fibers, they innervate the extrafusal fibers. And the gamma motor fibers, they innervate the intrafusal fibers. So now, the extrafusal fibers, these are what are involved in the tone of the muscle, and the intrafusal fibers are involved in the reflexes, the spinal reflexes, such that if you have an upper motor neuron lesion, here it's saying on the tone, it's called on the tone in the upper motor neuron you have hypertonia. But how does hypertonia come about? So on the upper motor neuron, we said if there's a lesion on any of these tracts, then it means the upper motor neuron will be disconnected to the lower motor neurons. But of great importance, I want us to look at this, pay attention to this nuclei here. They are located in the medulla oblongata. They are called the medullary retrospinal nuclei. So these nuclei, they have fibers which run down here to the anterior gray bone. So these fibers, they are inhibitory. They help to inhibit movements on the lower motor neuron. So And as these upper motor neurons are running down to the spinal cord, they release, they have collateral fibers here which run to these majorly to the spinal tracts, and they release excitatory. So when an impulse comes here, it will stimulate the major articular spinal tract, but these guys will release an inhibitory movement here. So let's say you have a lesion in the upper motor neuron here. So it means these excitatory fibers won't be, excitatory neurotransmitters won't be secreted, won't be sent to this major articular spinal nuclei. So, meaning, if this major articular spinal nuclei, they are not stimulated, it means they won't inhibit the movements here. And what will happen is that since there is no any inhibitory movements, the extrafusal fibers, these alpha motor neurons, they'll be hypertonic, like they'll be hyperfunctioning, such that just a small neurotransmitter which will be released here, it will cause these fibers to be more tonic. This will cause hypertonia because there is no inhibition from the major articular spinal tract. Then, since there is no inhibitory impulses on the on the gamma motor neurons to the intrafusal fibers, so this will lead to a scenario that is called the hyperreflexia on the upper motor neuron lesion here. So it means the spinal tract, these people will be the deep tendon reflex will be hyperreflexive because there is no control on the reflex, because these are it's more like they are rendered useless because there's upper motor neurons are not working. So now an aggregate of what? Hypertonia and hyperreflexia is what is called spastic paralysis, because in spasticity by term, it's more like there's resistance to what? To movement. So that's what spasticity means. Then the opposite happens to lower motor neuron lesion. You have flaccid paralysis and an aggregate of flaccid paralysis is what? Hypotonia plus hyporeflexia. Just the same mechanism I was talking about here. But here the thing is, the upper motor neuron, they are intact, but the problem is here on the lower motor neuron. So it means even the impulses which they send to the extrafusal fibers and the intrafusal fibers will be low. So you expect less muscle tone and less reflex here, because there is no any neurotransmitters which send to these muscle fibers. So that's why the type of paralysis which is found in lower motor neuron lesion is flaccid paralysis, hypotonia and hyperreflexia, causing flaccid paralysis. Flaccid is something which is like floppy, something, which is, something like that. Then the next parameter is fasciculations. So fasciculations in upper motor neuron, they are absent. Upper motor neuron, they are absent. And what I mean by fasciculations is that more like involuntary, unwanted, jerky muscle movements, like rapid and involuntary movements, especially of the tongue and even the hands. So what would be causing that? Like, for example, in an upper motor neuron lesion, we said if there's a voluntary or a movement which is initiated from the cortex here, when impulses run all the way down here and they stimulate the lower motor neuron, then this neuron will secrete acetylcholine here. So in an upper motor neuron lesion, let's say something disconnects the connection between these upper centers to this lower motor neuron. It means there will be a disconnection between the brain and let's say even the muscles to the tongue. So that won't have much effect because the lower motor neuron will still be what? Producing acetylcholine, which will help to coordinate the movements there. No wonder the fasciculations, these unwanted jerky movements, they are absent in upper motor neuron, but they are present in lower motor neuron region. Why? Because when there's a lesion here, then it means acetylcholine won't be produced here, because it can be produced in the lower motor neuron. So if you have few acetylcholine here, then this cell will respond by upregulating the receptors here. Why? Because there's few acetylcholine, and these so it means there will be a lot of receptors on this cell, and these receptors will be hypersensitive, such that just any few acetylcholine which will be found here will do stimulate an intracellular reaction and will cause contractions, such that we're now causing these fasciculations. So these fasciculations, the take-home point is that just know that they come about because of less acetylcholine, and in a bit to produce acetylcholine will come from a lower motor neuron lesion. And fibrillations, these are just graphical representation of what? Fasciculation. So they will also be absent in what? Upper motor neuron lesion, present in lower motor neuron lesions. And what I mean by graphical representation is that those jerky movements, you get to display them on the EMG or the electromyography, so that you look at that electrical activity. So those now, the pattern that will be showed on EMG is what are called fibrillations. So if there are patterns of what fasciculations, you expect them to be present on the lower motor neuron lesion. Then the last two signs of these motor lesions, you have the Babinski sign and the Peter drift sign. So the Babinski sign is present on the upper motor neuron lesion and absent on the lower motor neuron. How? Because let's say these corticospinal tracts, they innervate and like let's say, for example, this is a foot. Then the muscles here, these are the extensors, and these are the extensors, and they help to plantar flex the foot. So this corticospinal tract will supply the what? Both the extensors and the plantar flexors. And under normal circumstances, the plantar flexors okay. So the upper motor neuron lesion here. So we said if there is a neuron supplying the foot, you have those supplying the extensors and the plantar flexors. So if there is an upper motor neuron lesion, and if there's an upper motor neuron lesion, it will affect more of the what? The plantar flexors more than the what? The dorsiflexors. Then this will lead to what? What is called the Babinski sign, such that when you stroke the foot here on the like on the bottom of the foot, you expect them to be plantar flexing because of the innervation, which is more on the what? The plantar flexors. But in an upper motor neuron lesion, the activities on the plantar flexors will be less. So this will cause, instead of the feet to be plantar flexing, there will be more of what? A dorsiflexing movement, and will cause a sign called the Babinski sign. So So for these signs and the parental drift, it's more like you outstretch your arms, then you close your eyes, then someone just try to push your arms downwards, then a bit. And if that force is removed, then still the arms, they are still going down. Then it will mean that the person has a positive parental drift, because they are unable to use their flexors to help them like raise their hands. And the same effect is here, like because on the arm, the corticospinal tract, they help to more of flex than to extend. So if the power of the cortex to flex the arm is less, then it means on the positive parental drift will be positive, meaning the arms will be moving down as of the flexing movement. So I think that's where like I wanted us to reach this far. I hope this explanation here was useful. I thought that was the best way I could explain. Yeah, unless there are any questions until this far.
No questions for me.
From the floor. I don't have any questions for my own, but I can say that in teaching, that the expectation that it really not the things or the not things that you know had. So do you have any info?
So I didn't get to. Okay.
I have a question. First of all, start with on the we talked about the broadcast area being responsible to help generate speech, and there's a point you raised, like you said, in CVA, which can be affected. So you know, people. Then you said also about people who are unable to produce the speech properly. They get irritated and agitated, and because it has a close relation to the prefrontal cortex, or something like that. I remember very well. Yeah. So my question is it really an effect of the relation of the prefrontal cortex with the broca's area in CVA versus, let's say, stammering, people who stammer? Because then they usually born with it. So I was thinking, is it really an anatomical problem between, for example, CVA, people who come with slurred speech and stuff like that? Can we say it's the same thing, or there's a difference in the pathological, in the pathophysiology of both?
Okay. Yeah, I think it's a good question. It has helped me to think otherwise. Is that I think on this aphasia, blockers aphasia, I think even from question, you are trying to like how maybe stammering is related to this blockers aphasia. Was that the question, or you mean like is it that maybe those people who stammer, they have a pathological condition or what?
Yes, that's the question. Is stammering also affecting the brokers?
Yeah. Yeah, so I can say, I can say the broca's area is underdeveloped in those people. Like, I think there are some people when they were young, they used to stammer, but as they grow, the stammering goes away. I think we have such kind of people. So it means yeah, sometimes it can even be a problem, like during fetal development, the brain development. There's just something that goes wrong with the development of the Broca's area. It can cause maybe those signs such that they are born with it. But most of the time, when you see those people who stammer, find that stammering mostly will go away as they develop or as they get older. Yeah, but for those who don't like this, where the stammering doesn't go away, I think maybe for that I don't know, but maybe it can be related to maybe an intraembryonic pathology that happened, but I'm not sure. It's just a speculation. Yeah. So I can't really say those people who stammer they have a pathological condition in the Broca's area, but I would say they have something that maybe it's still underdeveloped, such that because remember the Broca's area, it helps you to comprehend, like to speak fluently. So you can comprehend, those people, they comprehend the sound, they're able to understand, but they can't talk, like they can't make complete statements, and they are sometimes they even have long pauses to give a response. Yeah, it's because of the same effect. I don't know if I answered your question.
Okay, thank you.
Yeah, all right, thank you. Any other question? I think we don't have any questions. I think for me, I can hand over to the host. So I don't know again if this PowerPoint, but I think I'll send it. So for this scenario, I think it has answers here. So you can just go through it and just trying to update what we are discussing. But it's an interesting case. You can just try to read more about Phineas gauge. It will help us to understand more about the lobes, the frontal lobe especially.